So welcome back. I will start with the review of in the last class. I discussed about what is denaturation and what is degradation. Denaturation is the breakage of hydrogen bonds between um, between the two strands, and degradation is the breakage of phosphate backbone. Okay. Hydrogen bond breaking between two strands is reversible. That means if the strands are complementary, they can re-anneal. Okay, these are assumed these are the hydrogen bonds. So if the strands become separated, that is called denaturation. And when they base pair again, they become double-stranded, and that is called as re-annealing. And whereas degradation is something like this, where the DNA could be broken in one or both of the strands, something like this. One DNA is broken down in the middle like this, then that is degradation. You can also think of it as phosphate backbone is just broken on one of the strands, and whereas the other strand is fine. That is referred to as nick, N-I-C-K. Okay, so you have to remember that. Sorry. So what I have right now is in this one, I want to differentiate. What we have discussed also is about hybridization. So here we have two DNA molecules, wild type and another mutant, you can call it as, that has a mutation, you can think. And then these were denatured. These two strands have become separated. So are these two strands which became separated or denatured. And if you mix them up and allow them to renature slowly by bringing down the temperature gradually, you can form different types of molecules where wild type strands can become together and the mutant ones together can become. But sometimes you may have a mixture of molecules, one from the wild type and the other from the mutant. They can hybridize as shown here. So here you have two strands with different genetic information, and in that, if base pairing occurs between them, then you call that hybrid, right? And formation of that is called as hybridization. So we also discussed about what is hyperchromacity. We discussed that single strands absorb more at 260 nanometers. And to remember, 260 nanometers is where nucleic acids absorb maximum at, and 280 is for proteins. Okay. And you can calculate melting temperature. Melting point is the temperature at which 50% of the double-stranded DNA is, a DNA molecule, double-stranded one, has denatured to single strands. Okay, that is the midpoint where you see somewhere here. That's the temperature is the melting temperature. So when double-stranded DNA absorbs less compared to that of single-stranded DNA, because as we discussed earlier, in single-stranded, the ring structures and the side groups are more free to absorb more energy or the light from there. That's why you have higher absorption by single-stranded DNA, and that is called as hyperchromacity. We also discussed about the effect of salt. We had a discussion on what are the strands, what are the forces that keep these two strands together, what are the forces that are acting against being together. Numerous hydrogen bonds are the ones that keep the two strands together, whereas the negatively charged phosphate backbone, there is high electronegativity, so they are repelling. So in high salt concentrations, you would expect that there is reduced repulsion. Why is that? When you have NaCl, for example, Na plus binds to the negative charges on the phosphate backbone on both the sides. You can just put, I'll just put as plus. So the repulsion between these two phosphates is reduced. Therefore, only the attraction is working, right? Then you need more energy to break it. So that is how you can also think of it. And we also discussed that the GC content, the higher the GC content, the higher is the melting temperature. The simplest answer is because GC has 3 hydrogen bonds between them and not as opposed to 80 hydrogen bonds. We also discussed about DNA topology. It's about how DNA is organized in a three-dimensionally or a higher level of structural organization. So in that, we are not going into a lot of things, but I did share videos I would want you to watch. It comes into theoretical and abstract ways of thinking. So it's a good idea to have some things. So twist is any time one strand, I'm talking about one strand, I didn't say one double DNA molecule. One strand crosses over another strand. That's twist. What is right? This is the component. Right is one double-stranded DNA molecule crossing over or folding upon itself like that. That is one right. So every DNA molecule, every DNA molecule has twist. So if you had approximately about, if you are thinking it as one turn equal to, has about 10 base pairs. Okay, typically it is 10.5, but for ease of calculation, I'm saying 10 base pairs. That means in every turn you have one crossing like this, that is one twist. So if we have a molecule, DNA molecule of 100 base pairs, then the twist that it should have is 10, right? Because there are 10 turns. So one turn is equal to one twist. If you make this 100 base pair molecule have about 12 twists, that is supercoil. That is, it is more than required. And because of which, to release the tension, DNA forms structure like this, that is right. Right? So if you can have only 10 twists, but you're forcing the twist, it is a stress on the backbones. To release that, it folds upon itself, and that is right. I hope yesterday we had a small demonstration. Some of you participated. Thank you for those who have participated. I hope you were able to feel that the action of twist, right? And the cumulative of twist and right is called as the linking number. So we will not go into too much of details because this is the only thing that we will discuss here. But it is important for several things, especially when we are talking about topoisomerases and electrophoresis. So topoisomerases, there are two types of topoisomerases. There are two types of topoisomerases, topoisomerase 1 and topoisomerase 2. Topoisomerase 1 makes a single strand cut. Okay? And then we have, just give me a second, please. Okay, sorry. There is a topoisomerase 1 makes a single strand cut like this. Okay, that is I defined to you about a nick. What is a nick again? Nick is something, it is a single strand break here. That is a nick. And then it passes these strands around this and seals it, something like this. Okay, so when you say linking number, it is about twist and right. Now in this linear DNA molecule, right is 0 and twist is 1, 2, we'll count it as 3 and 4, something like this, 4 twists. Look at the molecule that has formed here. You have one crossing, 1, 2, 3, 4, and say something like 5. Just give me a minute. Sorry again. Yes, so the point here is that the linking number has increased by 1. Okay, topoisomerase is has added one additional linkage. Okay, I will talk, I will better clarify myself once again. So this is 1 crossing over, this is 2, and this will be 3, and this will be 4. Let me do the same here. I'll count this to 1, 2, 3, 4, and 5. So the twist has increased here. Right is 0. The DNA is not folding upon itself. So you have now the total. Linking number is the cumulative of twist plus right. Right is 0 and twist was 4 in this case, whereas in this one it is n plus 1. So topoisomerases are increasing. Topoisomerase 1, there is another topoisomerase we will discuss, topoisomerase 2. Topoisomerase 1 increases the linking number in steps of 1. Okay, why is that? Because it is making a single strand cut, it's making a nick, and then passing this strand here, and then sealing it up. How do these topoisomerases work? You will find many enzymes throughout this course. I would want you to know the molecular mechanism so you will be better, you will find a better sense in associating different aspects of molecular biology. So here is the topoisomerase 1 enzyme. It has a tyrosine residue. Tyrosine is a hydroxyl amino acid. Using that OH group, which is activated, it makes a nucleophilic attack on the phosphate backbone and makes a cut there. And this enzyme becomes kind of conjugate along with it. It's covalently linked with the one of the strands, like the phosphate, 5' phosphate of the DNA molecule here. And after the exchange or the second strand coming and all those, the twist increasing in twist of and by steps of 1. The 3' OH again makes a nucleophilic attack on the phosphate backbone, phosphate 5' phosphate of this strand, and then reseals it. There is no involvement or there is no input of energy. So if you want to talk about topoisomerase 1, you can think of it is simple, is that they make single strand cuts or nick. Second thing is they increase the twist by steps of 1 because they are making single strand cuts. The third one is that there is no requirement for ATP, not required. Okay, these are the 3 things that you would want to remember in case of topoisomerase 1. And then topoisomerase 2 will become much easier. Let's just compare with topoisomerase 1 and topo 2. Just looking at the picture, I hope you will be able to tell me what is happening here. Is it single strand cut or double strand cut? It is a double strand cut. Whereas here in topoisomerase 1, it is a nick or single strand cut. Second thing is that because it is cutting double strands, it will make increase or decrease. The change in twist will be in steps of 2. Okay, whereas in topoisomerases, it will be in 1 steps of 1. No ATP is required for topoisomerases, whereas in topoisomerases 2, there is a there is a requirement for ATP for the functioning of the topoisomerase 2 enzyme. So in different steps of molecular biology, as we proceed, we will learn the importance of topoisomerases in different things. But what all can it do is something you need to see. So here are 2 DNA molecules, circular. In the last class, I described what is CCC DNA. This is for covalent, covalently closed. And this is circular. Covalently closed circular DNA. So if I have a molecule like this, okay, I'll call it molecule A, and I'll call this way, and I'll have another DNA molecule like this, A, B, C, and I will have this. Okay, I'll have a series of questions. I encourage you to try to answer. Which of them are all circular? I want you to just tell me the options that are circular in nature in this case. D is circular, yes. C is not circular. Is B circular? At least you can type if you don't want to say it. Is B circular or not? B is circular. And is A circular? Am I audible to you? Can you tell me, please? Hello? Okay, good. Yeah, well, I'll continue then. So is molecule A, is it circular? No, it is not circular. If you see carefully, there is no difference between A and C. Only thing I arranged it same in a circular way, but it's actually not a circular one because there is a gap here. Second thing is closed. Because we already said the A and C are not circular, we will eliminate them. Which one is closed? This is not closed. There is a nick. And this one is closed. Right? And when I say closed, it's about covalently closed. Right? This is one phrase actually. Whether it is covalently closed or not. So because this is not, the molecule B is not closed, you call it as open. It is open circular DNA. Whereas molecule D is covalently closed circular DNA. I hope you understand what is difference between open circular and closed circular. Closed circular is there is no gap. The phosphate backbones in both the strands are continuous. If it is open circular, there might have been a nick as shown in molecule B. Okay, what can topoisomerases do to 2 molecules like this? Because it can make a double strand break, they can catenation. That is, they can link these two molecules together. Okay, and topoisomerases can also decatenate them, make them into two independent circular molecules. Topoisomerase 1 cannot do anything to in the case of A, because making a single strand break doesn't catenate. But if you have a molecule like this, in which there is a gap wherein you see a single strand region a little bit, then probably topoisomerase 1 can do. Why is that? Topoisomerase can only make a single strand cut. It cannot make a double strand cut. Similarly, topoisomerase 2, which plays a role, very huge role in majority of the um, chromosomes, it can separate the DNA molecules something like this. Or if there is a knot formed within a DNA molecule like this, it can make it into simpler ones like this as well. So I hope you will remember something like this. And I also discussed about electrophoretic separation. I wanted to tell you about what is relaxed and what is supercoiled, right? If you are having right and other things, when linking number, say for example, there is a 100 base pair circular DNA, okay? What should its linking number be? That should be twist plus right. Assume right is 0. That means DNA molecule is not in this state. It is just in kind of this state. And with twist is, because I said 100 base pairs, that you can expect 10 helical loops, helical turns, and that means the linking number is 10. But if you introduce more number of linking, more twist into it, then the DNA molecule, if I introduce more than 10 or less than 10, then say if you have 12 is the twist, then you call it as positively supercoiled. If I reduce this to say 8 twist, I am kind of unwinding it, then it is negatively supercoiled. Both negative supercoiling as well as positive supercoiling is a kind of potential energy. Okay, these are kind of theoretical. You watch the videos, we can always discuss it again. It's a kind of potential energy. So majority of the DNA is usually in negatively supercoiled. It is not in positively supercoiled. When we discuss about replication and transcription, we will get to why is being negatively supercoiled better than positively supercoiled. So DNA can exist in linear, or it can be in a relaxed form, where in this case, as I was describing, the linking number or twist is just a 10, or linking number is 10, because right is 0 and number of turns is 10, so we can say 10 twist. In this case, we do not have anything, it will still be the same. 10 is the linking number. Here, we linking number is 10, or it can be more or less. I will put as 8, but then you may have right, because it is folding, it is running on one, it is crossing over itself, something like this. In this case, if you reduce even more to link twist of 6, and you may have to increase the right, because you see 1, 2, 3, and 4. Right is every time a DNA molecule crosses onto others. So more supercoiled, the more compact the DNA molecule forms. The more relaxed, it comes to relaxed, then it will be in, it occupies more space in a way. So during electrophoresis, DNA moves from negative electrode to positive electrode because DNA is negatively charged. DNA is negatively charged because of the electronegativity that is present on the phosphate backbone. So all nucleic acids are more negatively charged, so they will travel towards the positive electrode. So you should have loaded somewhere in here. And we have to remember, the more supercoiled, the faster they would travel. Less supercoiled, they would have traveled lesser, and linear travels next fast, and the relaxed DNA is usually the slowest. Okay. So then there is something about Ethereum bromide. Say when we do electrophoresis, we cannot see where DNA is and where it is not. We will, the picture that is depicted here, it is taken using a camera in black and white, and that's why you see it as white bands. You call them bands. You would have loaded the DNA in somewhere here. When you see it, when you do it yourselves, you will find it nothing big deal about it. Okay, did I do anything wrong? Or yeah, that is fine. Negative electrode is here, and this is the positive electrode. So sample will be loaded here, and the charge will be applied, and DNA will move based on its size and conformation. Two things have an effect: size and conformation. Size is number of base pairs, right? Okay, something is, and the conformation is about whether it is supercoiled or relaxed. So in this picture that is shown where A, B, C, D are added, all the DNA molecules are supposedly the same molecular weight. That means they have the same number of base pairs. What is different? Whether they are supercoiled, conformation is different, whether they are supercoiled or relaxed. Okay, the topology is different. So to visualize these, DNA is not visible. It's say colorless when it is in aqueous thing. So you cannot make it out. To be able to visualize DNA, we use a molecule that is called as Ethereum bromide. Ethereum bromide, I hope you can see that it has 4 ring structures within it. You can also think of it as, so this is DNA molecule, right? Double stranded. This is a base and this is a base. You see, this is base stacking. And when you have Ethereum bromide comes in between and stacks along with the bases. Why is that? Because you see, very similar to that of bases, Ethereum bromide also has the same ring structures which are hydrophobic. So you can think of it like this. This is a base that is of the DNA. This is one strand. This is the other base with the planar structures like that. And you can think of it as the other bases are, like this. Ethereum bromide can just fit in here like that. And it is making hydrophobic interactions with these bases. They share the same hydrophobic rings, similar hydrophobic nature of the ring structures. So the red one that is shown here is all Ethereum bromide. So Ethereum bromide interacts with DNA. It is a non-covalent interaction. It is actually a hydrophobic interaction or stacking interaction. And so when you do electrophoresis, there are 2 ways of doing. Most people add Ethereum bromide in the gel, in the agarose gel, and we write it as Ethereum bromide. Ethereum is the one that has this. Bromide is the one that fluoresces and Ethereum bromide fluoresces and gives us a bright orange kind of color, orange pinkish type. We will see that, and you will know when you do it practically. So all you need to know as a part of theory is that Yttrium bromide binds with DNA. It's a non-covalent interaction. It is a hydrophobic interaction or stacking interaction. Why is that? Because Yttrium bromide has the Yttrium bromide has ring structures similar to that of bases, which are hydrophobic in nature. So Yttrium bromide can stack along with the bases like this. Okay. I have another, I wanted to also discuss another part of this same lesson, which we will do it now. This is about RNA. Okay. So RNA is something that is central to all of molecular biology. People believe that all of life has started with the RNA, because RNA has multiple things. We will get to it and probably discuss the same in a later class as well. RNA can act as genetic material, not in as only in viruses we see that. Some viruses have double-stranded RNA, some viruses have single-stranded RNA as their genome. That means there is a potential for RNA to act as genetic material. RNA acts as the messenger. If you recollect from the central dogma in molecular biology, the messenger RNA is only played by RNA. And RNA can act as an enzyme. This was kind of later found. So typically you have specialized roles as well. Sorry. So RNA can also act as enzyme. Those are referred to as ribozymes. Okay. So in most organisms or all cells, the genetic material is DNA. But there are viruses in which RNA can act as genetic material. All or most enzymes are made up of proteins and they are very efficient. But there are few enzymes wherein RNA can act as an enzyme. There are very few examples, and those are referred to as ribozymes. RNA is the messenger. That means in a hypothetical stage during the origin of life, the one molecule that could have required to perform all these things would have been the originator, which are later taken up by DNA as genetic material and proteins as enzymes. And that's the basis for the theory of RNA world. And the debate still continues. So for the moment, we appreciate the logic of why RNA world is more probable, but we never know. Quickly, there are differences between RNA and DNA. The first one is about 2', right? 2' OH. Ribose has 2' OH, whereas deoxyribose that is present in DNA has H in the 2'. That's why deoxy, one oxygen atom is missing from ribose in DNA. And the second one is about uracil. DNA has A, T, and G, C, whereas RNA has A, U, and G, C. What is the difference between uracil and thymine? So uracil is easy to draw. I'm not changing it, solving for the double bonds. But yeah, so you have, this is uracil. And if I put a methyl group, that is thymine, this is the difference. So 1, 2, 3, 4, 5. 5-methyl uracil is thymine. Okay, that I keep telling. So I'm doing that repeatedly so that you will become comfortable with bases. I mean, drawing the chemical structures of it. So this is the typical hydrogen bonding that you would see between A and U, uracil. Yeah, okay. It's the same repetition. Deoxyribose is this hydrogen, whereas hydroxyl group in 2 prime. Okay, this would be 1 prime. This is the 5 prime. And thymine, this is the difference. Yes. So in RNA, we are trying to see what are the differences. There are non-canonical base pairing. Canonical means what you would normally find or intuitively what is right. But non-canonical is something out of the normal. So normally you would see a U base pairing, but um, in RNA you can also find GU base pairing. Okay. Now GC is of course present, but you also have sometimes GU base pairing. Similarly, even more abnormal is a triple base pairing, UAA. So adenine and uracil can base pair in this way. I wouldn't insist on knowing all these base pairs, but the chemical structures of AT base pair and GC base pair is what you should know. Um, I will give an assignment shortly. You will have some time to present it. Maybe I'll post it tomorrow. Let's see. So typically RNA is single stranded, but you can have double stranded RNA structures forming either as part of viral genomes or even there are interactions between two different RNA molecules. Again, it's the same thing as base pairing. But then RNA, because it's not because there will be differences between different RNA molecules sequence wise, they will base pair wherever possible. They may remain as single strands if there is no homology or complementarity. Sometimes you may have partial homology. Something like this might become, those that do not base pair usually bulge out like this. You may also have, there is no base pairing in this whole thing at all. So they might form a loop like this. And in some cases, a RNA molecule can base pair by itself like this. And this structure is called as a hairpin. So there are different structures that are possible in RNA that is based on the complementarity of the interacting bases. Okay. So there is another structure that forms, it's a complex structure, tetraloop. If we can try to track it. So here is the phosphate, and then this is the single strand, and this I'm trying to trace the phosphate backbone along. Okay, so it is forming. There is an interaction between this phosphate and C. Okay, there is something else also. There is an interaction between uracil and this ribose, and then you have the base pairings. Okay, so I don't need you to mug up this, but you should understand that tetraloops are present and they have some biological significances in giving stability to the RNA molecule. So there are other forms of structures like the pseudoknot. One second. Okay, yeah. So in pseudoknot, it is a single RNA molecule. Okay, this is single RNA molecule because of base pairing, because of homology or complementarity. It is intramolecular hydrogen bonding. Why do I say intra? Because it is single RNA molecule. If there were two different RNA molecules, they are interacting like this, then you can call it as intermolecular hydrogen bondings. That means there is hydrogen bonding between two different molecules. Whereas in this case, it is one single molecule folding upon itself and base pairing. So the first step, you have base pairing here, and I hope you recognize the structure. It is a hairpin, right? You see this, and there is a loop here. And this again can base pair with some of the bases from this part, that is this one. They can interact with these bases from the loop. And then it forms a structure like this, and which is referred to as a pseudo knot. Yeah, we will discuss the rest of the things in the next class, but I would want the processing of tRNA and other things I will discuss when I'm discussing translation. It will make much more sense now, because I'm assuming you're entering into reading core molecular biology. I want you to have fundamentals, and then gradually build on to more complex phenomena in molecular biology. So, but I think you know about RNA, tRNA. I'm not bothering you with what is tRNA and all. Just try to, as part of what I want you to appreciate is this structure alone. That to allow you to know that RNA can form very complex structures, unlike DNA, and there are many differences. So I want you to also have RNA can form structures like this. This is a one hairpin. Again, this is another hairpin, and so on. RNA is able to form three-dimensional structures, stable structures. That means if there is a structure, there is a function. And when you have complex structures, you have complex functions or multitude of functions. And that's why people believe in RNA world, or they tend to have, that's the uh, most likely hypothesis at this stage for the origin of life. Not only that, they also have modified structures in their modified bases. This is dihydrouridine, and psi is referring to pseudouridine. Okay, there are methylations in some places of the bases, and you have modified bases and so on. So different RNA molecules have different modifications on them. They can form different structures, which increases a complexity, and thereby giving rise to multitude of functions. It's something like this. You can always think of biology or a cell. I hope you know the differences between prokaryotic cell and eukaryotic cell. Uh, the there are, say, 10,000 different types of cells, molecules in it. If there is a structure, it will interact. It has a property based on its side groups, its shape, and so on. Based on that, it will interact with one or other types of molecules. So the more complex structures a molecule can form, or more complex structures that are present within a cell, one would expect that there are more number of interactions. So structures will give rise to interactions between these structures, will give rise to functions or phenotypes. Okay, structure will have a function, and that function could be because of charge, that is because of side groups, or because of shape, and that will give rise to interactions, and interactions will give rise to you can think of phenotypes. Okay, if there are two different structures, A and B, if they have interacted, there will be some change, and that change is the phenotype. Either A will change, or B will change, or together they will change another molecule C. Now think of a cell which is a continuous and dynamic structure within which there are different structures and thousands of interactions and thousands of outcomes. Okay, and that is where whichever has a good outcome will survive or exist, and that is the basis of all biology. Okay, If the interactions are bad, then the cell would die. If the interactions are beneficial, then the cell will survive. That is something like applying Darwinian evolution even at the level of molecule. You call that chemical evolution. One would wonder how a structure of tRNA has formed. One can also wonder how the structure of ribosome has formed. In my own understanding, I still have trouble understanding how the ribosome has evolved because it's the most complex thing. I do not wonder about how a DNA can give rise to another DNA molecule, or I do not wonder about how DNA can give rise to RNA, because they are all in the same language of nucleic acids, right? So at this stage, I find it easy and simple or logical that that is what should happen, and it's very simple. But translation is much more complex because you have a molecule in DNA, say, and then RNA is produced. From RNA, you are producing a protein. From the language of nucleic acids, you're coming to the language of amino acids, which is something amazing. And that is done or carried out by ribosome. How can this happen? And it's a very complex process. You need a lot of tRNAs to bring the amino acids, and you need mRNA that is coming out of transcription, no issues. And how do you read the mRNA? That's the difficult one. And how do you make these proteins, and that the in a proper sequence like this? Those are some things that are amazing, and that's why to me ribosome is something still unsolved to myself in understanding about the evolution. How did ribosome evolve is a big question to many other researchers as well. Okay, if you have any questions, I will stop here. um, I'll stop here. You can ask me if you have any questions now.